This is the fourth mini lesson in the series, Protein Structures and Analysis in Biotechnology. So, methods of protein analysis is what we will be talking about in this mini lesson today. So, there are a number of standard techniques that scientists use to analyze protein. A couple of these are protein quantification, chromatography, and polyalkylide gel electrophoresis, or PAGE. So, let's get into what each one of these are. So, protein quantification, we can see scientists often need to determine how much protein is in a sample. So, we can use a colorimetric assay for this purpose. These assays involve adding chemical reactive dye to a protein solution. The dyes change colors in the presence of a protein. The intensity of the color change is directly proportional to the protein concentration. The relation, we can once again see Beer-Lambert's law come back to this. So the relationship is based on Beer-Lambert's law, which states absorption is directly proportional to the concentration of light, being a light absorbing substance in a solution. So we can use a spectrophotometer to measure how much light is being absorbed by a specific sample at a specific wavelength. These spectrophotometers work by shining a narrow beam of light through a sample in a cuvette. And these are clear containers with a standard 1 centimeter light path. Measuring how much light passes through a sample using a detector, these spectrophotometers can then determine how much light is being absorbed by the substance. Protein concentration is determined by creating a standard curve using known protein concentrations, measuring the absorption of an unknown sample, and then comparing unknowns to the curve to find their concentration. So there's 3 different common colorimetric protein assays that we'll talk about. We have Buret's test, Lowry's assay, and Bradford's assay. Each assay varies in sensitivity and chemical compatibility. So Buret's test is the oldest protein concentration test, and it was first used in 1833 by Ferdinand Rose. It involves copper ions in an alkaline solution reacting with the peptide bonds, and these produce a violet-colored complex when proteins are present. This solution absorbs light at 540 nanometers. It's not highly accurate or sensitive, so it's rarely used for precise quantifications. But it is helpful to detect the presence or absence of a protein, so it's just a qualitative test. We have Lowry's assay, which was developed by Oliver Lowry in 1951. This is one of the most influential scientific methods. So Lowry's paper has been cited over 300,000 times. It is the most cited scientific paper of all times. So how this works is it takes copper ions to treat the proteins, and then the proteins will react with fluorine calculor reagent to produce a color change. The resulting solution will absorb light at 750 nanometers. Due to the sensitivity and reliability for measuring protein concentration, this is one of the highest used assays in industry. Then we have Bradford's assay, which was developed by Marison Bradford in 1974. It uses dye Cosamin Brilliant Blue, or G250, and it interacts mainly with the adenine and other specific amino acid R groups. And how it works is in the absence of proteins, the dye is reddish brown and absorbs light at 750 470 nanometers, the presence of proteins, the dye suddenly changes to a blue form, and the color change indicates protein binding. Bradford's assay is fast, sensitive, and once again widely used in the laboratory. So now that we can quantify the amount of proteins, we can look at protein process or properties that we can use in analysis. So each protein has a unique amino acid composition and gives it a unique property. So structure dictates function. Electrophoresis or chromatography are two techniques that we can use to separate proteins by exploring their differences in physical properties such as size and charge. So, are they positive, negative, neutral, things like that. So, first, we're going to talk about protein size. Unlike DNA, which is measured by base pairs, protein size is measured by mass. This is due to the varying size of amino acid R groups. The unit of measurement that we use to measure proteins is called a Dalton. A Dalton is approximately the mass of a proton, 1 and 1/12 of a carbon atom's mass. Most proteins, are greater than 100 amino acids long, and we measure these in kilodaltons. For example, tryptin, titin, a muscle protein, is approximately 3 million daltons, and thromyosin is about 5,000 daltons. Protein size is useful for separation and analysis. Scientists often use relative molecular weight instead of molecular mass when referring to protein sizes. So, protein charges is another way we can separate out proteins. Proteins are made of amino acids, and these amino acids have different charges. The net charge of a protein depends on the pH of its environment. So, isoelectric point, or PI, or IEP, is how we're going to measure what the protein charge is. The pH at which the protein has no net charge is going to be the PI. Each protein has a unique PI. So protein charge and pH. How are these related? So if the pH is lower than the pH which the protein has no net charge, the protein then has a net positive charge. If the pH is higher than where the protein has no than the pH that the protein has no net charge, the protein has a net negative charge. The principal key for techniques like isoelectric focusing or ion exchange chromatography is going to be looking at those protein charges. So I just want to reiterate, PI is where the pH of a protein has no net charge. So let's talk about different methods that we can look at these proteins in. So protein mass spectrometry is a highly sensitive method for determining protein mass. It can be used with pure samples and complex mixtures, and it is widely used in a biotechnology research lab. This is essential for biomanufacturing of protein, for protein analysis and quality control. Mass spectrometry is a powerful tool for identifying and characterizing proteins. It is best for determining protein mass and amino acid sequences. It can also be used to study protein's 3D structure, post-transitional modification, and protein interactions. Some of its advantages provides detailed data not available through other methods. There are some challenges with protein mass spectrometry, right? We need pretty large, intricate instruments to require or to run a mass spectrometry. We also need special training So some lab solutions can be shared facilities or outsourced services for analysis in mass spectrometry. Um, Unfortunately, this is not a machine we have. I did get to use one in college, and it was pretty awesome. We can do chemical mass spectrometry too, and it is great if you have access to the machine. So, protein mass fingerprinting. This is where proteins can be analyzed by intact by digesting proteins. We can, but digesting proteins into fragments is also common before analysis. So, protein mass fingerprinting process is where proteins are broken down into fragments using something called protease, which is an enzyme that breaks down protein. So protease examples would be trypsin. Trypsin cuts after lysine or adenine, except when followed by proline. And then we can take our masses, so our broken down fragments of protein, and run a mass spectrometry analysis. So peptides can be analyzed by processed to analyzed to produce a spectrum showing the mass of each fragment. And then we can take data comparison and compare the experimental spectrum to a database of known or predicted protein sequences to determine what protein it was. We can use computer programs to detect peptide fragments and generate a theoretical mass spectrum. We can also use protein electrophoresis, very similar to DNA electrophoresis. So, since proteins vary in size and charge, they can be separated and visualized using electrophoresis. There are some challenges. Proteins are much smaller than DNA fragments, and they require tighter matrices for separation. So we use a polyacrylamide gel electrophoresis or a PAGE. Right, so we use this instead of the agarose gel that we make for DNA. Proteins are separated based on size and charge in the polyacid matrix. So, a couple of things on this. So we can look at protein structure complexity. The chemical structure of proteins are more complex than that of DNA, so it's going to separate out a little bit different. We also have net charge variation. So unlike DNA, which is uniformly negative across all DNA molecules, each protein has a unique net charge, right? We looked at if the pH and the pi were different, they would have different charges. This results in some proteins migrating towards an anode and others migrating towards a cathode, or a positive and negative sides of the electrical field. We also can see shape influence the protein's movement within that PAGE gel. So proteins have different shapes due to their tertiary and quaternary structures, and these shapes affect how proteins migrate through the gel. So there are a couple of different things that we can do to make this easier and move through this gel more uniformly. So first thing that we can look at is using sodium docyanine sulfate, or SDS, and it's a detergent that coats proteins and gives them an overall negative charge. The degree of charge is proportional to the molecular mass of the protein. Additionally, SDS can cell lyse, so it breaks open the cell membranes for them to release their proteins. It can also denature, so it can partially denature proteins, disrupting their 3D shape, making it easier to move. Then we can go and heat the sample. So heating to 90 degrees Celsius fully disrupts that 3D structure to make them somewhat lay flat and move easier through the gel. So We can then add reducing agents like diethanol or martocurthol to break those disulfur bonds between the cysteine amino acids. So after the SDS and the heat treatment, proteins pretty much are linear and negatively charged chains that migrate through the gel based on size rather than net charge. So most of the time we'll see in those biotechnology labs, we're going to run an SDS page for these reasons, right? So what is so special about um, polyalkyl gels? So the gel structure are thin polyalkyl gels, about 1 millimeter thick, and they are cast on two transparent plates. We use a vertical electrophoresis chamber, um, and these pass an electrical current from an electrode into a buffer reservoir at the top of the electrode chamber to the bottom of the gel. Um, it typically runs at 200 volts for 30 minutes to an hour, and it runs until the dye front reaches the bottom of the gel. We can see TGX gels be run at 300 volts for 20 minutes for faster result. The same power supplies for vertical gel electrophoresis gel electrophoresis can be used in SDS pages. So these gels are made by polymerizing acrylamide in the presence of a biacrylamide, a TEMED, and an amino prosulfate. This polymerization is a chemical reaction that makes this polyacrylamide gel. It is non-remeltable, so unlike the agarose gel that we make for vertical chambers, the polyacrylate gel cannot be remelted down once it's polymerized. So once you have made it, it stays. Once you have made it, it stays in that shape and you cannot reuse it. So since this is a pretty caustic substance, acrylamide in its monomeric form is hazardous and can cause cancer and nerve damage. Polymerized acrylamide, as in the page gel, is safe and used in products like diapers and for moisture absorption. So if you've ever opened up a diaper, those little gel things, when they get wet and soggy, that's what this is, polymerized acrylamide. So precast gels, we can buy commercial page gels, and these are often used to avoid the hazards associated with polyamide gel preparation. So the percent of the acrylamide in the gel affects the separation of proteins by size. So small proteins, the best percentage to use is a high percent gel, so 15%. Large proteins, we're going to use a low percent gel like 7.5. And then we have gradient gels, and we can see these used in a wide range of protein sizes. Like when we investigated wide range, we're going to use a gradient gel. A gradient of acrylamide percentage is used to optimize separation. So when we run electrophoresis, we need a buffer, right? We use a TEA buffer for vertical. Well, in these SDS pages, we're going to see tris, and this is a buffer that helps maintain the pH when running. Of the running buffer, SDS, like we've talked about, and this keeps the proteins negatively charged and denatured, and then glycine, which helps resolve proteins into tight bands during this electrophoresis. In preparation, we take TGS, which is commonly available in a 10x solution, and dilute it to 1x before using it into our SDS page. So, problems with this discontinuous buffer system, we can get fuzzy bands. So, in a regular system, proteins are loaded at the bottom of the well uh, of wells, and they enter the gel before those at the top, leading to unresolved protein bands. The solution to this problem is the discontinuous buffer system helps address this issue by creating two buffer regions. So stacking the gels help concentrate proteins before they enter the separating gel, and then separating gels ensure better resolution and sharp protein bands. So we do stack two gels on top of each other. We have a low percent stacking gels is a low percent of acrylamide at 4 to 5, and they compress proteins into narrow bands. And then we have a resolving gel, which has a high percent, which allows for the separation of proteins based on size. So the difference in pH and molarity between the running buffer and the stacking gel causes the proteins to compress and stack at the intersection. Result, all proteins enter the resolving gel at the same time, leading to a sharper, more defined band. We can see visual indicators, so migration of protein size standards and dye front help track protein stacking, so we can see them separate out. So gradient gels and SDS pages, we have no separating, stacking, and resolving cells. The acrylamide percentages increase continuously down the gel. So it starts from 4% and moves to 20% at the bottom. Some advantages of this is it provides a wider range of protein separation, and it allows for small and large proteins to be separated at the same time. It is ideal for analyzing samples containing proteins of various sizes. So, why do we want to see these proteins on these gels? So, why visualize them? Most proteins are not visible with the naked eye, right? In the gel. So we go through and we visualize. So visualization is essential for quantitative, what's there, and qualitative, how much is there analysis. So we can have different methods for visualizing proteins once they've been on this gel. So the total protein stain, so we can use a silver stain or a Kosomin blue, and then we have a specific protein stain, and these can be antibodies or tags that go in and target a specific protein. We also have stain-free gel technology, which is a newer method that allows for fast, sensitive detection without traditional staining. So the most common one is going to be the Kosomin Brilliant Blue, and it binds to the specific amino acid chains, and it stains those proteins blue. The gels are stained, and then the excess dye is removed, so the only thing that remains stained are the bands, the blue bands on a clear background. Other stains would be silver stains, and this is extremely sensitive, and it detects even the smallest amount of protein. Then we have fluorescent stains, which require specialized equipment, but it offers high sensitivity and multiplexing potential. Stains differ in sensitivity and equipment requirements and are chosen based off of the experimental needs. So that stain-free gel technology works like a traditional SDS page, but with a key advantage. It contains a reagent that reacts with tryptophan residue in the proteins. Upon UV exposure, the proteins fluoresce, making those bands visible. Some benefits to this is there's no staining or destaining required. Results are faster and with less handling of the gel. It enables real-time visualization and documentation, and it is ideal for saving time and simplifying the proteins analysis workflow. So other types of page, so beyond the SDS page, it can also also be used to separate proteins and other molecules based on physical properties at the same time. So versatility, acrylonized gel are adapted for analyzing charge differences, isoelectric points, and configuration in binding interactions. These specialized separation techniques are used in protein purification, protein to protein interaction studies, and analytical biochemistry research. So, a native page is a separation of charge to mass ratio. Unlike an SDS page, native pages separate proteins based on their natural net charge to mass ratio, not just size. So no SDS, no denaturing. Samples are not treated with SDS. They remain folded and functional, and it's useful for studying protein complexities and activities. A key advantage of this is maintaining protein structure and functionality. Proteins can be recovered from gels and used in downstream applications.